To start the demyelination process, antigens are needed. These can be obtained either from foreign organisms that mimic actual proteins, or by using proteins from your own central nervous system. Today we'll be talking about the latter. To start, myelin basic protein is citrulinated by PAD, which adds an arginine group to it. Conversely, myelin proteolipid protein is acylated by CAT, which adds an acetyl group to it. Since PLP and MBP help compact the myelin sheath, changes in their structure will damage the myelin integrity and make the proteins more vulnerable to breakdown. The broken down residues, or full proteins, are then exocytosed via the oligodendrocyte or permeate through the damaged membrane. These antigens are then taken up by the lymph node after traversing the blood-brain barrier, and by microglia in the CNS. Here, antigen-presenting cells like B cells, dendritic cells, and macrophages process the antigens. APCs then express these antigens using major histocompatibility complex class 1 and 2 to the naive CD8 and CD4 T cells, respectively. This exposure causes the T cells to differentiate into different types, depending on cytokine exposure. This is the first activation stage, and to continue, they must be reactivated. To do this, the T and B cells leave the lymph node and migrate until they find the antigen. The T and B cells manage to penetrate the blood-brain barrier via binding to ICAM1 and VCAM1. Here, they interact with microglia, astrocytes, and macrophages to reactivate, though cytotoxic T cells can bind to the antigens expressed on oligodendrocytes to reactivate. It is possible that free-floating antigens can bind to the immune cells, and this is typically how B cells are activated into plasma cells. Following activation, plasma cells release antibodies that specifically bind to exposed sites of MBP and PLP. Complements then bind to the antibodies and are cleaved, signaling to immune cells to release cytokines and for phagocytes to come break down the membrane. Complement cleavage also causes membrane attack complex to activate, leading to further membrane degeneration. Both TC1 and TH1 release interferon gamma and tumor necrosis factor alpha to encourage further immune cell differentiation and increase cytokine production. TH17 does much in the same, though only signals to TC cells to produce more cytokines. TC17 also encourages further immune cell differentiation. Interferon gamma and tumor necrosis factor alpha cause macrophages to release reactive nitrogen species and reactive oxygen species. These cause lipid peroxidation, which disrupts membrane structure and fluidity, and also damages DNA. Once the demyelination process ends, phagocytes endocytose the myelin fragments and remyelination may be attempted.